The Cell Cycle by Henry
and Jake.
The cell cycle is one of the most fundamental aspects that separate living organisms from non-living organisms by allowing the continuity of life to exist. The cell cycle is composed of two main parts, interphase and the mitotic or M phase. Interphase dominates 90% of the cell cycle, even though the most important stages occur in mitosis or meiosis. Interphase can be broken down into three subphases, G1, S, and G2. In interphase, DNA replication occurs, producing two sister chromatids joined at a region in the center known as the centromere through cohesin proteins. This leads into the M phase, consisting of either mitosis or meiosis. Mitosis is the formation of two daughter cells with diploid chromosomes. Mitosis can be broken down into 5 stages. The first phase is prophase. In prophase, chromatin condenses into sister chromatids, which are joined by a centromere. Nucleoli disappears and the mitotic spindles begin to form from the microtubules of the centrosomes, which begin to move away from each other. The second phase is prometaphase. In prometaphase, the nuclear envelope begins to disappear and the mitotic spindles attach to the kinetochores of the chromosomes. The kinetochore is a region of specialized protein located at the centromere region. As the spindles attach, they begin to move the chromosomes back and forth. The third phase is metaphase. In metaphase, the centrosomes have moved to opposite poles of the cells and have lined the chromosomes up along the metaphase plate. The metaphase plate is an imaginary plane in the center between both poles of the cell. Next is anaphase. In anaphase, the cohesin proteins are cleaved, allowing the sister chromatids to be pulled apart towards each end of the poles. At this point, each chromatid is known as a chromosome. The cell now begins to elongate, preparing for the last stage of mitosis, telophase, and cytokinesis. In telophase, two daughter nuclei form and the nuclear envelope begins to reform. Now the chromosomes begin to decondense, becoming invisible once again. Cytokinesis finishes off the cell cycle by dividing the cell into two daughter cells with diploid or 2N chromosomes.
After mitosis comes meiosis. Meiosis occurs in eukaryotic organisms and involves the development of gamete cells. In males, this is sperm, and in females, they are egg cells. Meiosis is split into two parts, meiosis I and meiosis II. In meiosis I, the cell starts as a 2N cell. Meiosis I starts with prophase I. In prophase I, the chromatin condenses and DNA is exchanged between homologous chromosomes through crossing over. This results in a creation of alleles, which are different forms of the same gene. Chromosomes are also held together at an area where the crossing over occurs, which is called the chiasmata. The paired and replicated chromosomes are called bivalents or tetrads. After prophase I is metaphase I. In metaphase I, the homologous chromosomes move together and line up on the equator of the cell. The microtubules from the chromosomes attach to the kinetochores on the respective chromosomes and pull. After metaphase I comes anaphase I. In anaphase I, the spindle fibers attached to the kinetochores are shortened and pull apart the homologous chromosomes. The two sister chromatids are pulled apart into each half of the cell. After anaphase I comes telophase I. In telophase I, the sister chromatids arrive at their respective poles. Two daughter cells have half the number of chromosomes, but each one is made of two chromatids. The microtubules disappear and the nuclear membrane reforms. After telophase I, cytokinesis occurs. However, DNA replication does not occur. The second half of meiosis is meiosis II. Meiosis II starts with prophase II, in which the nuclei disappears, the chromatin condenses, and the centrioles move to opposite poles of the cell. After prophase II comes metaphase II, in which the sister chromatids move to the equator of the cell and spindle fiber attaches to the centromeres. After metaphase II comes anaphase II, in which the centromeres are cut and the sister chromatids are pulled apart by the spindle fiber. These chromatids move toward the opposite poles of the cell. After anaphase II comes telophase II, the final step of meiosis II. In this step, the chromosomes uncoil and the nuclear envelope reforms. By this time, there are 4 daughter cells, each with N chromosomes. Mitosis and meiosis have a few differences. Mitosis ends up in 2 daughter cells, both of which have 2 N chromosomes. Meiosis ends up with 4 daughter cells, each of which has N chromosomes. In males, this ends up to be 4 sperm, and in females, it results in 1 egg and 3 polar bodies.